I titled my talk, It Just Keeps Getting More Fun. It'll come up in a minute, hopefully. Yeah. Because that's how I feel about this whole cell-free nucleic acid area. You know, in my career, I tend to get bored easily, and I switch fields every 10 years or so. And this is one I haven't left because it's just been really fantastic, and it seems like there's always something new and neat coming up. And I think you've got a flavor of that from all the talks in this session. What I'm going to talk about today is much less clinically oriented. Some of the more sort of earlier basic science things we've been doing in my lab over the past year or two, and giving you a sense of what things might be working their way in the clinic several years down the road, and giving you the sense that I think there's still a lot of interesting things to do in this field. Here's my conflict of interest slide. I've been involved with several different companies. As one wise tech transfer officer once told me, no conflict, no interest. And I think more seriously that the work doesn't stop once you publish the paper and get the editors and referees satisfied to really see impact in the clinic. It often requires partnering with folks outside of the ivory tower, and you've seen some examples of that earlier today. So I first got in this field a little more than a decade ago when we figured out how to develop the first non-invasive prenatal test for aneuploidy based on applying these what were then emerging techniques of high-throughput sequencing to the phenomenon of cell-free DNA. And it kind of took off incredibly quickly. It was quite a lesson to me. After we published our paper in 2008, a number of clinical trials were launched, both in academia and industry. They read out about 3 years later, and that marked the launch of a number of commercial tests. There's a handful of companies in the space right now, and sort of cumulatively across the different ways of doing this. And by 2013, 500,000 women received a non-invasive prenatal test, doubled the next year. By 2016, it was 3 million women in that year. And people who began studying sort of folks who were offered both a non-invasive test or an invasive test saw that the use of invasive tests were decreasing rather dramatically. And so this is something that has sort of been absolutely a blast and a joy to see that translation can happen on a pretty reasonable time scale and affect the lives of large numbers of people. I didn't do any of that translation. What we were doing was different things. And as Mickey mentioned in his talk, we got interested in applying cell-free DNA to organ transplantation. And we're doing a study on that. And my postdoc, Ewan DeVlamick, noticed that some small fraction of the cell-free DNA he was sequencing was not human and was in fact due to the microbiome and the virome, and he realized that in our cohort we could study sort of how your microbiome and virome respond to immune suppression, and a very interesting story came out of that. Mickey was sitting right next to him, realized, oh, we should be looking at infectious disease. That led to Carius getting spun out of the lab. What Mickey didn't tell you was what he was working on before he did Carius, and it was he was helping mentor a graduate student named Mark Kowarski, and they were interested in the sequences that don't map to anything. Okay, so the way this game sort of works is you purify cell-free DNA, you put it on the sequencer, you get a bunch of reads out, and then you have to map those reads to a database. And you can map them to the database of all known genomes. You can look at all sequence reads, anything anyone's ever found. And you find that about 1% don't map to anything that anyone's ever seen. And we got curious about those and began to think about how to assemble them and what they might be deriving from. And over the course of a number of years and different studies, we had sequenced more than 1,500 blood samples from pregnant women, heart transplant patients, lung transplant patients, people with cancer, healthy people. And that added up to about 37 billion reads. And Mark decided to analyze, and Mickey decided to analyze the entire data set and try to take out everything that mapped and assemble what was left. They did that and got something like 7,000 contigs that were bigger than a kilobase pair. And as we try to classify those, it appeared that a small fraction of them were closely related to known ones, and for whatever reason it slipped through the filtering. A few million were divergent, so they were kind of related to known organisms, but were sort of new strains or things that hadn't been seen before, but about 10 megabases of them. Sorry, this is sorry, not reads, but assembly size. So about 1 million bases of divergent. About 10 megabases of assembly were completely novel, just didn't map to anything in the databases, appeared to be actually real signal, not artifact, because they had genes in them. We could take those sequences, PCR them back out of the blood samples for a fraction of them that we checked, and just things that people had never stumbled across before because they were maybe in parts of the body that were never being sampled, things that have never been cultured. And as we dug more deeply in them, there's about 3,000 of the reads were in this category, of the contigs were in this category. And as we try to classify them, what's the closest possible relative that's ever been seen before? Some of them, were not that interesting, novel species, but many others, at the level of phylum, kingdom, and not mapping to anything, indicates that there's just an enormous amount of diversity within all of us that had never been seen before until one combined sequencing with cell-free DNA. I'll give you sort of one example around the sort of more divergent ones to give you a sense of kind of the scale of what's going on. So this is anelloviruses. This is the tree of anelloviruses. These here in the white area are the ones that have human hosts. The bold ones are new ones that we discovered. The not bold ones that were there before. The green are the ones that are non-human hosts. These yellow ones are a whole class of divergent ones we discovered, which in phylogeny are closer to the non-human host anelloviruses than to the human ones. So a really divergent new set of anelloviruses just waiting to be explored. It's really kind of amazing and fantastic. So all of this work has been looking at DNA, but not all viruses are DNA-based. Some are RNA-based, and we'll get the question, how about RNA viruses? We began working on that. In particular, Wenying Pan, who's a graduate student, was working on sequencing cell-free RNA from pregnant women through the course of pregnancy, and that led to a bunch of very interesting results, which we'll come back to in a minute. But she, at one point, began to look at the non-human RNA in those samples and was able to see that there's a prevalence of sort of the expected human viruses there, and you can read them out by mapping to the known genomes. It's been more challenging to try to discover new viruses that way for technical reasons that I'd be happy to address in the discussion. But here's another example of one where this woman had a very high level of parvovirus. This is one that the doctors were really interested to know about because it can affect health. This one sort of cleared on its own without treatment, and you can see that the levels went away over time going postpartum. But it just gives you the sense that it should be possible to look at RNA viruses as well through liquid biopsy type measurements. So there's DNA, there's RNA. How about modifications of DNA? There'll be a session later this afternoon about epigenetics, and you'll hear from one of the real world experts, and Joe Ecker, about epigenetics more broadly. I want to talk about it in the context of cell-free DNA, and in particular about the work of Chun-Chao Song, who was a postdoc in the group, who joined me after doing his PhD at Chicago with Chuan He, who was another one of the world leaders in epigenetic measurements. And what Chun-Chao had done as a student was develop a way of looking at hydroxymethylation of DNA through next-generation sequencing. And so I think you're probably familiar with methylation. You've heard it. It's the fifth base. It's a way to turn off DNA. But when you want to turn the genes back on, you have to demethylate the DNA, get it back to its normal chemical state. And there's a number of intermediates in that process. And one of the most important intermediates is the hydroxymethyl state. So it goes methyl, hydroxymethyl, a few other things back to unmethylated. And it's useful in the context here in that it's sort of capturing something changing in time. You're capturing the genes that are being turned on again, that have been silenced and are being turned back on. And so in my group, Chun-Chao developed a way of doing these sorts of measurements based on very small amounts of DNA, such as you get from cell-free DNA. There's really limiting amounts. And he came up with a very nice way of doing enzymatic labeling followed by click chemistry so we could pull down on beads the hydroxymethylated fraction of cell-free DNA and then sequenced that. And he proved that it was a useful analytical assay. And then he began looking in cancer patients. And I'll give you a couple of little vignettes about that. In the first, he was looking at pancreatic cancer patients. That's a very challenging one. There's not a great, it tends to be discovered late. There's not a lot of symptoms, and so it's often, when discovered, too late to treat. He found very strong signals in the parts of the genes that were hydroxymethylated versus not, and could separate healthy from cancer patients with a very strong signal, and even down at the level of individual genes, ones that make sense in the course of disease, were giving an important signal. And sort of buoyed by results like that, he went and looked across about half a dozen different cancers. And what he noticed when he began doing a sort of dimensional reduction of the entire hydroxymethylome in these patients is that they tended to cluster together by cancer type. Okay, that's healthy there. That's breast cancer there in green, which doesn't really resolve. But all the other cancer types more or less cluster together. And we did a lot of work to convince ourselves that this wasn't due to batch effects. And it sort of suggests that this sort of approach, looking at the epigenetics of the cell-free DNA, solves what's been a major problem in liquid biopsies in cancer, which is that you've got a tumor, but you don't know where it is. And this may be providing information to help tell people where to look. A lot more research needs to be done. There's a very small number of samples, but we're very encouraged by it and looking forward to see what's going to come in the next few years. In the last few minutes, I'd like to return to cell-free RNA, which I touched briefly on a moment ago, and tell you about some work we've done, first in collaboration with Tony Whisker-Ray, to use cell-free RNA as a generalized tool. Eric and I like to call it a molecular stethoscope, to measure the health of any given tissue in your body. And we realized that if you want to measure the health of any tissue, cells that are dying under stress, you should look for transcripts that are specific for that tissue in the blood and monitor their change in time. And we had done this in a few different contexts and then decided to team up with Tony Whisker-Ray, who had been collecting and banking plasma samples from patients with Alzheimer's, mild cognitive impairment, and normal, age-matched, very carefully curated. And we began to do PCR assays for neural-specific transcripts. In particular, APP, which is the transcript for the protein that causes the plaques. We could see that as you average over all the patients in the group, there were elevated levels for those with Alzheimer's relative to normal, and intermediate levels for those with mild cognitive impairment, suggesting that this may be a handle into getting a blood test that would diagnose neurodegenerative diseases. We did larger numbers of genes as we increased the size of the panel to 40 transcripts and started to create classifiers. We began to see more and more separation. This is reduced to a two-dimensional view. If you do it with a larger number of genes and create a classifier, you get what looks like a reasonable receiver-operator curve. Again, the numbers are small. There's a lot more work that needs to be done, but this is, for us, a very encouraging sign that that one day this sort of liquid biopsy will be generalized to all sorts of tissue health, including the brain. I have a few more minutes? Yeah? Okay. Last example, returning to something that's been just a really interesting area to work in has been human development. And we wanted to use cell-free DNA to monitor human development by looking at placental and fetal transcripts through the course of human pregnancy. And have been doing this in sort of a low-resolution way for a while, and eventually teamed up with, and we've been doing this funded by the March of Dimes, and Dave Stevenson runs a really phenomenal center at Stanford. We're part of that group. And we then eventually connected with Mads Melby, who runs the Danish National Biobank, and he's got a sort of fantastic IRB over there, which I'll show you in a minute, to do high-resolution studies through the course of pregnancy, work that was done by Tweenago and Mira Mufarej in my group. So what Mads did was get his IRB to agree to consent women to give blood every week during pregnancy. So these very brave women came in and were giving blood every week. Mads is doing a variety of studies on these. He was willing to share with us some of these precious samples to do cell-free RNA studies. And what we did was put together a PCR panel of genes, which I'll show you in a second, to try to see if we could really track development of the fetus. And it looks sort of like this. Each of these panels is a different gene. The samples have been binned a little bit in time for signal-to-noise purposes. And the color coding of the gene names here are, these are placental genes. These are immune system genes, so trying to monitor the maternal immune system. These are fetal tissue-specific genes here. And you can see what happens generically. Placenta is the easy one. It's a huge tissue, changes dramatically over pregnancy, that the expression goes up over the course of pregnancy, and then after delivery goes away. That's the important control there, and you can see that in all the fetal and placental ones, the signal is going away after pregnancy. Some of the curves are going up dramatically. Some are plateauing. There's one in the immune system plateauing. There's one plateauing from the fetal tissues. And you can see that it should be possible to put these together to try to build a clock that, having measured all these genes, you should be able to tell where the fetus is in, for example, gestational age. And so Mira did that with the data, and it actually works pretty well. Here's the training set, here's the validation set, so these weren't used for the training. And you can look at, if you ask, when is the blood drawn, it's not so great in the first trimester, but it's a lot better later on in pregnancy. This ends up, estimating gestational age being an important clinical problem in a variety of contexts. Ultrasound is kind of the current standard, but that's only useful in the first trimester. It doesn't provide a good signal if you don't make the measurement in the first trimester. And in developing world, it's not an available instrument, whereas PCR is a relatively inexpensive thing to do and is much more universally used. So if you can do a PCR test to estimate gestational age at any point in pregnancy, there's clinical value in that. And so this is just showing that with one blood draw, we're not quite as good. With two, if we use two blood draws, we're about the same as ultrasound in terms of accuracy. So I think I'll stop there. I'd like to leave some time for the panel. I just sort of summarize by saying this has been a great journey. Every time I think the field is done, some new amazing thing happens, and it's sort of like the gift that keeps on giving. So not ready to leave the field quite yet. Thank you.